Bismillahirrahmanirrahim. Another video on inheritance and genetic engineering, and these are mostly questions from the paper 2 questions, not the MCQs. Now, coming to the first question, it's four definitions of terms used in genetics are shown in table 5.1. Definitions: the outward appearance of an organism. Well, for each of the definitions, select an appropriate term from the list and write it in the box provided. So you have to do this. The outward appearance of an organism, I always say to remember it as pH, physical appearance. So phenotype, we say this is a brown, this is a black furred rabbit, this is a white furred rabbit, this is blood group B, because you can actually test somebody and say, okay, well, he's got blood group B or blood group O. A length of DNA that codes for a protein, a length of DNA that has to be gene, it can't be anything else. That is the definition of gene. So actually it's the definitions which were asked in the syllabus, and then now they put it in another way. Having one set of chromosomes, that is haploid. Having one set of chromosomes, haploid, is usually the gametes, and these gametes are produced by meiosis. Then type of nuclear division which gives daughter nuclei that are genetically identical. Now that has to be mitosis. So, I mean, just going through the other ones which we have not been asked, what would be the statement for genotype? The genetic makeup, which is always expressed in letters, like this would be genotype. Then mutation, a spontaneous or a random change in the DNA is called a mutation. A change in the DNA which leads then to an altered protein being formed. An altered protein being formed. So I'm just giving you a revision a bit of it. Then coming on to the next part of the question, which is the B part of the question. A couple who have blood groups A and B have 4 children. Each child has a different blood group. I usually teach this to you in the class. Use the space below to draw a genetic diagram to show how this is possible, and use the symbols IA, IB and IO for these alleles. So this is the line that is given to you, parental blood groups. Then the next is parental genotype. So this will be IAIO. And this would be IBIO. You see, if you said IAIA and you said IBIB, then the offsprings could only be IAIB. But they have said that each child has a different blood group. So this was not possible. I've done it for you just to make you understand why that was not right. So this is the only way you can get 4 blood groups is by writing IAIO and IBIO. And then now the genotypes, gamete genotypes. Gamete genotypes is just you write these separately and you write them at a distance. So IA, IO, IB, IO. But you have to understand is why did this sort of, you have to write these separately, the IA, IO and IB, IO. And then it says children genotypes. So this would be, if you connect, I always say you can number this 1, 2, 3, 4, and you start off with 1, 3, 1, 4, 2, 3, 2, 4. So 1, 3 would be what? IAIB. 1, 3 would be IAIB. This is 1 and this is 3. So 1 and 3. So 1 and 4 would be IAIO. 2 and 3, this one here and this one here. 2 and 3 will be IBIO. And then 2 and 4 would be IOIO. So this would be blood group AB. This would be blood group O because, sorry, A because O is recessive, and this would be blood group B, and this person will be blood group O. You see, the question, it said each child has a different blood group. So in order to get four blood groups, you'd have to do this combination. Now I'll just revise this with you in another way so that you can understand this. So you can see the possibility that the person blood group A is IAIA and the B is IB, even then it will be only IA. So there will be only one type of blood group in the children. All the children, whether they're three children or four children, all would be blood group AB. Possibility. Now what will the children be? So children can be IAIB. So this is blood group AB. And the child can be IAIO. And the person is blood group A. The other two are the same, AB and AO. So there are only two possibilities. Children can be, but in the question it said four blood groups. If the question had said two blood groups, then of course this was correct. This you could have done this. But this question was each child has a different blood group. So that is why the answer to this was different. Now the part C of the question says explain what is meant by codominance. You may refer to the genetic diagram and B to help with you with your answer. Now I give you a very simple example. I say red flowers are crossed with white flowers and all are pink flowers. So there are what? Three phenotypes: red, white, and pink. Now this is a phenomena of codominance. CR and CW. And here we don't write capital R and small r because they are both dominant, means both show their effect. Like if you take red paint and mix it with white paint, you'll get a shade of pink. So red into white, pink. So a blood group A and a blood group B, and the children are blood group AB. So it's codominance, which means that there are two alleles and both are expressed, meaning both are dominant. And in the heterozygous, they are both present. Like here, the red and the white, all the offsprings are pink. So similarly, the blood group A and the parent is blood group B and the children are AB, meaning the A has its effect and B has its effect, and the blood group is called AB. It's not called A or B, it is AB. So this is a phenomena of codominance. So how would you word this? You word it like this: there are two alleles, both are equally dominant, or you can say both are expressed, and that will result in a third phenotype. So in the heterozygous individuals, the blood group AB is an example. So this is how you have to word it, 3 marks, but you have to be careful how you word these answers. This is a very important thing which some of you are struggling with. So you have to realize is that you have to memorize certain parts of the syllabus and certain parts of the questions so that you are very clear when you write it, you get your marks. You might know it. Some of you say, yes, I know it, but I don't know how to write it. But I'm sorry, you don't know it then. If you don't know how to write it, how do you say you know it? Now, coming to part D of the question, insulin produced by genetically engineered bacteria first became available in 1982. Before 1982, insulin had been prepared from dead animal tissues, dead pancreas, animal slaughtered, and then the pancreas removed, and then the insulin extracted from that. Explain the advantages of using insulin produced by genetically engineered bacteria rather than insulin from dead animal tissue. So this is a question which actually is checking whether how much of the, why did genetically engineered insulin come into being, is that you actually use this insulin to treat diabetes. Now, insulin that is produced by genetically engineered bacteria is the same as humans, while the ones we got from dead animals like cows, that was cow insulin. So many people were allergic to the cow insulin, just like people are allergic to different foods. Some are allergic to fish, some are allergic to nuts. I'm allergic to mangoes. So, I mean, there are people who are allergic to anything, and they could cause a very, very severe allergic response when you give them cow's insulin or pig insulin. And then, of course, we had religious issues. Muslims did not allow pig insulin to be used. Then, of course, the animals had diseases, and those diseases could be transmitted to humans because we were using animal products. then genetically engineered insulin can be improved. We could change the structure of it slightly and make it better. And then, of course, we did not, when we used bacteria, we stopped slaughtering animals and taking out the pancreas out of cows and pigs. And then, of course, it's a much cheaper and more readily available because you can produce large scale on a large scale, however the need may be. It's according to the supply and demand. If you need more of it, well, you can produce more of it. But in the other case, we used to slaughter animals. So that was really, really very difficult and expensive. Then the fact that bacteria reproduce very quickly. So you've got 1 million genetically engineered bacteria. So in 20 minutes, there are 2 million, and then 4 million, and then 8 million, and then 16 million. And then the trouble is that the more and more people are suffering from diabetes. So the need for insulin is increasing. So just helping you to learn the wordings that we need to write the answer to this question. Insulin is used to treat people with diabetes. People will not be allergic to the genetically engineered insulin. Animals are not killed. It is cheaper and more readily available. Don't say it is easier. That's a reject. Because it's not easy to genetically engineer bacteria. It's a very long and tedious process. So don't say easier. That was a reject. So many students must have written easier. Then the fact that bacteria reproduce very quickly, every 20 minutes they reproduce, so that is a very good way of uh, producing insulin on a very large scale. Then genetically engineered insulin can be modified. It can be changed slightly to make it more effective or to make it long acting or short acting. And those are the things that we talk about in modification. Now, coming to the part E of the question, figure 5.1 shows some of the steps involved in genetic engineering of bacteria. This is a human cell. This is a bacterium, chromosome nucleus. Here you can see we've removed the chromosome. We're cutting out the insulin gene from the human cell. And then here we've taken out the plasmid. And here we've added a restriction enzyme S so that we've cut the plasmid. So the plasmid was complete and now it is cut. And then we have joined the insulin gene here. This is the insulin gene. We have joined this with this, and we have got recombinant DNA or a recombinant plasmid. Then we have put it into a new bacteria, and then these bacteria are reproducing and they are producing insulin for us. Now, the question they've asked you, name structure R. Structure R is a plasmid. And what is it made up of? So there are two marks for it, you can see. There are two marks, and it is made up of DNA. So name structure R plasmid. And what is it made up of? You could have said DNA. Then state what is added at stages S and T. S and T. S, what did we add? We added a restriction enzyme. And at T, what did we get here? T, we had the human insulin gene or the human insulin DNA. So it said here in the question, state what is added at stages S and T. Okay, so I hope everybody gets this. What is this question? What is added at stages S and T? So this is another restriction enzyme, 1 mark. And at T, we added a human insulin gene. Okay, now we read through this question, and I won't attempt at doing it. I'll just read through it, and you can all have a look at it and try to do it yourself before you look at the video. A species of plant has white flowered plants and blue flowered plants. If a homozygous white plant was crossed with a blue plant, all the seed produced with only blue flowers. State which flower color is controlled by the dominant allele and explain your reason for the answer. Use the symbol big B, small b to represent the two alleles for flower colors. State the genotype of each parent plant. Blue plant, white plant. State the genotypes of the offspring. Draw a genetic diagram to predict the likely results of a cross between one of the blue offspring and a white flowered plant. Now please pause the video here. I'm going to just scroll it so that you all can do it again. How do you attempt this question? It's very simple. It said white is crossed with blue and all the offsprings are blue. So what does it tell you? It tells you blue is dominant. Why? Because if it said white into white, all white, then that was recessive. But it said white into blue, all blue. So that means no, though the white allele is there, now the white allele is small b, and the blue allele is big B, big B. So all the blue are big B, small b. Now, how do you figure that out? Because it said the white flower is homozygous. And if we say white is recessive, so recessive has to be two small letters. Homozygous white means it is a recessive trait. And the blue, when all were blue, there were no white ones. Because if we had, I'm just giving you an example, if we had this into this, then we would have some white, but we do not have any whites. So this is what we have to understand is state which flower color is controlled by the dominant and explain your answer for this answer. So it was very easy. You said blue because no white flowers are present in the offspring. So in the presence of the blue allele, the white does not show its effect. Like for instance, the white here we have the blue, the white is for small b, but we don't show its effect. So blue, no white in the offsprings. Use the symbol B and small b to represent two alleles of flower color. State the genotype. Genotype means letters. So the blue flowered plants will be what? Will be capital B, capital B. The white will be small case b. Then the genotype of the offspring will be heterozygous. So this is homozygous dominant. This is homozygous recessive. This is heterozygous. Then draw a genetic diagram to predict the likely result of a cross between one of the blue offspring. So this is the blue offspring and a white flower. So this offspring into white, and white was homozygous recessive. White was this. So you complete the genetic diagram. Now in the genetic diagram, please remember, one of you asked me this question on WhatsApp. You don't have to, it's 4 marks. You just have to do it per line. So you write this separately, B, you write this separately, you write this separately and this separately. And you can circle it, you may not circle it, but you just have to write it at little space. And this is now the gamete. So this gets you 1 line, 1 mark. And then you may not cross, you may make these lines, you don't have to do these. But if you want to, you can. If you don't want to do it, don't do it. And then and the same here. So what do you have? This is blue. This is also blue. And this is white. And this is also white. So, and what is the ratio? It's 2:2. Or you could have simplified it and it was 1:1. So it's up to you to write it 2:2 or 1:1. Both were correct. You got a mark for that. So it is marked per line. So there were 4 marks. There's 1 mark for the ratio. So you got a mark here, 2, 3, 4, 5. So any 4 out of these, and you got your 4 out of 4. Now coming to part C of the question, figure 3.1 shows a cob of a maize plant, grains containing seeds. Figure 3.2 shows the length of the cobs. So the length of the cobs, we are talking about this, the length of the cobs formed by a number of different maize plants. All the plants were grown from seeds from one original cob. Now you look at here, what do we have here? Number of cobs. And what we have here on the x-axis is the length of the cob in centimeters. So there were some 7 centimeters, some 8, some 9, some 10, some 11. But the larger ones were, most of them, the greater ones were 12 centimeter. The number of cobs were in abundance. So if you look at this graph, you just, I always say, just join the ends, and then you see it comes something, and then you see here and here and here. So it's generally a graph which is something like this, a bell-shaped graph. Now this is always in continuous variation. Continuous variation shows there are two extremes, and then there are lots of intermediates, like the height of 16-year-old boys, the height of 18-year-old girls. So you were given this graph to look at it first of all, and then look at the data, just like I said, okay, length of the cob in centimeter, number of cobs. And then it says, explain the evidence visible in figure 3.2 that shows that this is a continuous variation. What is this? Shows extremes and all intermediates. Shows extremes and all intermediates of cob length. And that got you your 1 mark. Then it says, suggest 3 environmental factors that might affect the length of the maize cob. So the amount of light or light intensity, then the amount of minerals in the soil, the amount of water in the soil, then the temperature. So these are the environmental factors. More light, more photosynthesis. More minerals means more nitrates, more phosphates, more sulfates in the soil, so more growth. More water means, of course, the plant needs water because there's transpiring. So there should be no water. The more the water in the soil, more the water is going to rise up and come out as transpiration. If there is no water in the soil, then the stomata close and photosynthesis also stops. Then it says the last part, explain how the type of variation shown by the maize cob differs from that shown in the blue and the white flowers. In the blue and white flowers, it's discontinuous variation. There are only two types. There are no intermediates. We didn't say dark blue, light blue, and then some other purple and some other. We didn't say any other color. We just said blue and white. That's it, two types. Discontinuous variation means only very few categories, 2 or 3 or 4. Like we have 4 blood groups. There's no intermediates. There either you're a blood group A or a B or an AB or an O. That's it. There is no fifth or sixth blood group. We can't say in Iceland there are people who have blood group X or blood group Y or something like that. Or they have blood group C and D as well. No, there is nowhere on the face of this earth. Everybody is either A, B, or AB or O. That's it, 4 blood groups. So when you look at the environmental conditions in part 3, how is it going to affect the growth of a plant? The mean is, how can environment affect the growth of a plant? Yes, it can affect if there's less light. One plant is grown in more light and one plant is grown in less light. Or maybe there's some trees and they're shading some of the plants. Or there is a building nearby and that is shading the plant. So amount of light, then the minerals in the soil. Are there more? Are there less? Are there enough nitrates, phosphates, sulfates in the soil? Then is there enough water in the soil? And what is the temperature like? Is it warm or is it cold? Maybe one part of the area where some of the plants are growing, there's a warmer area, and some are in an area which is in the shade, so their temperature is less. Then third part was flower color, only blue and white, or you said no intermediate color. Thus, it is a discontinuous variation. There are only two categories. There are only two types of flower, blue and white. So this is discontinuous variation, because you have to understand it was from the previous question previous part of the question, which was on variation, types of variation, continuous and discontinuous variation. So that finishes this video, and I hope this is going to be helpful. If there's any problems, please do let me know, and thank you very much.